have DNA inside of us that serves as instructions to make us who we are. Think of this DNA as a document, and imagine one sentence in this document is missing a key word. That sentence would be unreadable. To fix this sentence, we would need to find a way to insert that missing word. In the Johnson lab, this is exactly what we're aiming to do with DNA. There are over 10,000 different diseases in which a single functional gene is missing. Due to this unreadable sequence, the body doesn't function as it should. If we were to insert a healthy version of this gene into an individual's DNA, they could be cured. But unlike a document, we cannot just insert a gene anywhere in the DNA. We need a safe location. A location that, if edited, would not result in any adverse consequences. Toward this aim, I'm evaluating a specific DNA location that could be safe for gene insertion. This location determines if you have a positive or negative blood type, and is called the RHD locus. For someone with a blood type A negative, for example, the gene at this location is already not functional. Yet, individuals with a non-functional RHD gene are completely normal. Because we know that this DNA location can be disrupted without causing any problems, we hypothesize that the RHD locus can be a safe place for gene editing. To insert a gene into DNA, though, we first need to make a cut in the DNA. To do this, we are using the CRISPR-Cas9 technology. CRISPR-Cas9 includes 3 parts: Cas9, which is a molecular scissor that cuts DNA, a guide RNA that guides Cas9 to a specific location, and a repair template that repairs the break in the DNA with the gene that we are inserting. With the help of my lab mates, I constructed these 3 CRISPR components. We made our guide RNA match the DNA found at the RHD locus so that it can tell Cas9 to cut there. Our repair template contains a gene called GFP that glows green so that we can visually prove when it is successfully inserted. I then added these 3 CRISPR components to human blood cells and confirmed that the GFP gene was not only inserted at the correct location, but also functional, as observed by this green glow. My next step is to repeat this process, but instead insert the gene that is responsible for the bleeding disorder hemophilia A. If successful, this innovative technique of using the RHD locus as a safe place for editing our genetic document can be applied to curing several diseases. Thank you.
Reimagine your future with a graduate degree from San Jose State University. Located in the San Francisco Bay Area and ranked #1 most transformative university by Money Magazine, San Jose State offers more than 100 high-impact advanced degree programs. You can conduct hands-on research in oceanography or wildfire science, develop new occupational therapies for human wellness, create cutting-edge digital media art, and so much more. Start designing the future you want today. Explore your options at sjsu.edu/graduate programs.
This competition gave me those tools such as how to present yourself, how your body movements make the audience think about your words.
A lot of grad students don't have this opportunity, and so this is a great privilege.
This experience was actually one of the most eye-opening experiences I've had at San Jose State because it allowed me to just understanding the breadth of work that's happening at SJSU. It's opened my mind.
It was very difficult to judge. There were 10 amazing presentations, and each of them was so different, and they each had their own strengths.
It's not easy to get your scholarship down to a 3-minute presentation like that. And so you have to start out of the gate very strong, and you have to hook the audience on something interesting about your topic, why it's important, why we should care.
It's not enough to have your ideas hidden in a research paper in a library somewhere. You have to get it out into the public so that people understand the value of the research that's going on. So we're developing activities like the Grad Slam as a way of helping students achieve that skill set.
Having this opportunity is going to help me so much when I eventually have to defend my thesis.
I would love it for our industry neighbors to participate in Grad Slam in a variety of ways. Perhaps they could serve as sponsors, perhaps they could send people to serve as judges. We would love them to attend. This is a great way for them to see some of the talent that's emanating from San Jose State University.
That moment of hearing your name get called, it's wonderful. All of the hard labor that I put into this come to fruition.